We'll get started now. All right, well, welcome back from the break, and thank you for coming back. Alexei, thanks you too. But I would like to introduce, yeah, he has coffee too. I'd like to introduce our next speaker, Alexei Tivanski from University of Iowa, who's going to tell us about chemical microscopy of individual submicron particles.
Yep. Thank you. So first, I'd like to thank Kim and Vicky for organizing this excellent symposium, and also all the previous speakers. They basically did all the work for me. So I don't really need to do a lot of introduction. I'm going to talk mostly about using different examples of complementary microscopy techniques towards single particle quantitative analysis. So my group is primarily physical and analytical, and so we're developing a variety of methods. And I would like to give you a few examples of different techniques and how we utilize them to study aerosol particles, specifically those that are related to sea spray aerosols and contain different types of inorganic and organic components. Primarily, I will focus on X-ray spectrum microscopy or scanning transmission X-ray microscopy, something that Vicky mentioned briefly, and how it can be combined with atomic force microscopy to learn some new information, and briefly micro FTIR microscopy, and also some examples work with collaboration with Alexander Laskin that we utilized SEM with EDX to learn something about the properties and compositions. I would like to point out, which is very important, is that all particles that I'm going to talk about today, they were substrate deposited from atomized solutions, and they impacted on different substrates. This is the graph that we already have seen, and it's really just underline the importance, and Kim really mentioned it very, very nicely, that there is a very large uncertainty in how aerosols affect climate and in terms of whether it's a negative or positive. So predominantly we assume that this is a negative effect that leads to the cooling of the atmosphere, and really the error bars are huge. And one of the things we're trying to do is to develop approaches, both experimental and techniques, how we can decrease such uncertainties. So I primarily will focus on particles that are on micrometer or less in dimensions. And so there are a variety of effects, such as direct and indirect aerosol effects due to the presence in the atmosphere, effects that include visibility, complex heterogeneous chemistry. And if you think about all these effects, there are a lot of factors that contribute, such as water content, how particles uptake water, how it depends on size, shape, density of particles, elemental chemical composition, mixing states, if you have external versus internal mixing, and so on. All of which are, as nicely presented by Alan, also depend on the relative humidity in the atmosphere. And there needs to be a nice quantitative, and I really would like to underline quantitative methods that allow you to study and measure these properties, ideally on a single particle basis. So the X-ray spectrum microscopy technique that we utilize in our group is based at the Advanced Light Source, Lawrence Berkeley National Laboratory, and the technique is called scanning transmission X-ray microscopy. So very, very briefly, the technique is based on the following schematic. You have monoenergetic X-ray beam, you focus it on the substrate, you have a substrate in the focal plane, you raster scan the substrate, you have particles deposited on it, and you record the intensity of transmitted X-rays. So you can quantify the absorbance, where I0 measured on sample-free region, and it can be related to this X-ray equivalent of a Beer's law. It has a very nice spatial resolution. The additional thing that we added for the first time was to do these measurements in the presence of water vapor so that we can do a variety of different humidity-dependent studies. So we can measure, for example, hygroscopic properties, but also study differences in reactivity or changes in the chemical composition as a function of relative humidity, for example. So for all these experiments, the substrates were deposited on silicon nitride windows. The spectroscopy that is used in conjunction with this method is near-edge X-ray absorption fine structure spectroscopy. So it's basically excitations of electrons from co-states to different unoccupied energy levels. So we can obtain both spectrum and the spatial resolution. So very quickly, data acquisition modes. You can do images under a single energy as you raster scan the sample. You can then repeat the collection at the various energy by repeatedly scanning the sample, so you can get a stack from which you can extract individual image spectrum. So here's an example, for example, carbon KH spectrum. And one of the things I want to point out here, and this is something that is specifically utilized in my group to study atomic compositions, is this difference between what we call post-H and pre-H. So this is for the carbon KH, but the same is true for oxygen or nitrogen and so on. So this difference critically only depends and only proportional to the number of atoms at the corresponding absorption age. And so that signal gives us information on how many carbon atoms or oxygen atoms we have. And so that leads me very quickly to the experimental approach that we developed, how we can study hygroscopic properties of individual particles. So we will utilize very simple sodium chloride as an example, and also look very quickly at the halide series. So the idea is to initially take a look if we can use optical detection to record the presence of vapor. So the answer is yes. So by varying the relative humidity, we can record the spectrum. We can quantify total oxygen absorbance. Again, signal directly proportional to concentration of oxygen atoms as a function of relative humidity. So we have very nice linear correlation. And so then we look at the particles, first of sodium chloride at the low relative humidity where they're dry. So we can look at the oxygen pre-H, post-H. We can take the difference, and there is no oxygen present, there is no water present. And then we can repeat this kind of measurements by raising slowly relative humidity, allowing the system to equilibrate, and repeating the same experiment. And if you look at the oxygen map changes, including also the pre-H energies for the oxygen, so right before the deliquescent phase transition, so when the crystal undergoes phase transition, becomes a droplet, we observe no oxygen. Immediately after we have the oxygen, we can quantify the corresponding oxygen absorption. And if we now go to the dehydration cycle, so going all the way up until we reach a certain relative humidity and now starting to decrease, we can study the efflorescence phenomena, as Alan also nicely described. And so in this particular case, somewhere between 48 and 44% relative humidity, we observe efflorescence. And as you can see, the particle completely loses oxygen. So what is critical here to point out is not only the fact, which is fairly straightforward, that we can determine relative humidities at which phase transitions occur, but we also have ability to measure differences in size. But most importantly, we can measure the amount of oxygen present by directly recording the optical density that corresponds to the concentration of oxygen atoms, and therefore the amount of water. And this is the basis of the model that we developed in the conjunction with these measurements, which basically looks at the cross-sectional analysis, records the optical density corresponding of total oxygen along the main axis of different particles, records them as a function of relative humidity, along with the full width half max. And the model that we developed allows us to connect these experimentally measured quantities with actual mass of oxygen in units of kilograms, without any knowledge of what type of particle, what type of composition we have. And so we can also scale it up and determine what is the mass of water or mass of oxygen. So let's take a look at a couple of very simple examples that illustrate that it works. And so this is what we did for a series of sodium chloride, bromide, and iodide. And as you can see, we can very nicely follow hydration and dehydration. Hydration in blue, dehydration in red. We can see very nice shifts in the deliquescence and fluorescence as we change the halide series. So this is nothing really new. What we also wanted to do, we wanted to double check whether our measurements of mass of water. And again, I would like to note that we are using optical means of detection to determine the mass of water, whether or not this is actually reasonable and accurate numbers. So for that reason, we wanted to switch from inorganic sodium chloride to something that we can actually spectroscopically detect. So sodium chloride, unfortunately, is not something that we can directly quantify using X-ray microscopy, because we primarily probe carbon, oxygen, and nitrogen. And so for that reason, we switched to another atmospherically relevant system, sodium nitrate. As you can see, it has, as part of a molecular formula, it has 3 oxygen atoms. So we can utilize that as a means to determine water to solute ratio. And that now we can plot as a function of relative humidity using exactly the approach that we developed. And this is what is shown here for the hydration in red, dehydration in blue. And now water to solute ratio is something that we can quantitatively compare with other literature measurements. So including elevated particles, for example, measurement from Tang and Fang. And as you can see, without any additional feed, so we just overlay their data with our data, we see a really perfect overlap. And that to us was a very strong indication that our measurements and our optical ways of detecting the mass of water of individual particles is accurate. Finally, we compared the values of deliquescent relative humidity and efflorescence relative humidity on the substrate deposited particles with those that are accepted in the literature. So we really observed a fairly good correlation. So it doesn't appear to be any substrate effects, at least for the systems we looked at. So this was one example. And the next, we extended it to something that was a little bit different. This was the example where we looked at the ketoenol tautomerism of fine malonic acid particles. And so we looked at the chemical transformations that occur as a function of relative humidity. Just to give you an idea, if you look at the malonic acid particle, if you look at the organic chemistry textbooks, you'll find that the keto form in the solution is by far the most dominant form. What we, however, showed, and I will give you very briefly experimental evidence for that, is in fact that when we have our deliquescent malonic acid droplets, the equilibrium shifts to a very significant way towards the NO form. Why it's important? Well, it's important in part, you can think about it from a reactivity perspective. The NO form is much more reactive relative to the keto form. And so this is a very important reaction. This is a new type of chemical reaction that occurs really at the deliquescent malonic acid particles at the levels where you have a very small amount of water. And so you have very high ratio of concentration of acid molecules to water. So very quickly, so we looked at a few individual particles. So this is a 2% relative humidity, 90% relative humidity for malonic acid. So we determined, just as I described, water to solute ratio. So we see very nice overlap between our X-ray measurements, and it overlaps with, we also did micro IR and also literature. So we have a very nice representative particles. And so what we did next, then we looked at the changes at the carbon and oxygen KH spectra of that very same particle at different relative humidities at 2, 50, and 90%. And so what you see here, it's a little bit a convoluted plot, but basically what you see is the following. As you increase relative humidity, you start to observe an increase in the transitions that correspond to C double bond C, COH, and the corresponding decrease in COH and CH. So this is what you observe at the carbon, and the corresponding changes are also present at the oxygen KH. And so if you think about it overall in terms of the corresponding changes, you will come up with this keto-NO type of the reaction. And so what we observe basically is that as you increase relative humidity, we observe a consistent shift from the keto to NO form. Now, one of the advantages of X-ray microscopy is the quantitative nature. So every single transition is proportional to the concentration of corresponding functional groups. So we can use the intensity, and this is what we did, of double carbon and COH at the carbon and oxygen KH as means to quantify the ratio of these two forms and actually determine quantitatively the equilibrium constant. So before we did that, we wanted to make sure that the effect we observe is not due to the X-ray damage of kind of a phenomenon. And so for that, we did a less destructive micro-IR analysis. And what is shown here is the different spectra where we took a spectrum at different relative humidity, relative to 1 and 2%. And basically, we observe the negative changes that are, one more time, consistent with the keto-NO form. So CO, CH2, and so on. So combining the carbon and oxygen KH, and again, the intensity for C double bond C from carbon and COH and oxygen, you can quantitatively determine the equilibrium constants. And this is what we did. And as you can see, we have at 2% dry conditions 0.18, at 50 1.1, and at 90% 2.3. So if you go to the bulk, that number is ranging between 10 to the minus 7 to 10 to the minus 4. So we observe a very, very significant shift. And what we argue, again, as I mentioned initially, this is due to the fact that we have a limited amount of water molecules that are solvating our acid. What is also interesting to point out is that in principle, as you start to increase the amount of water, eventually the change should reverse, and you should go back to the bulk conditions. So this is something that was very challenging for us experimentally because it's very difficult to go much beyond 90% relative humidity. So the other example that we developed was the idea how you can combine X-ray microscopy and atomic force microscopy to determine density of individual particles. So this is something that is very useful to assess, for example, how porous different particles are. And we can do it, again, by taking advantage of ability of determining absorbance using X-ray microscopy and measuring heights of particles using atomic force microscopy. The fact that we can utilize the very same substrates and very same particles using two different microscopy techniques basically allows us a direct way how we can experimentally measure this and H, and so that gives us a way how we can directly quantify from the fitting the density. And so that's what we did. So this is just a nice cartoon of the AFAM that I will skip. So here's an example, sort of a proof of concept. Looking at the sodium nitrate particles, so this is the example of how the particle morphology looks like using AFAM, and this is the corresponding for nitrogen and oxygen maps using X-ray microscopy. So we can run the spectrum. Everything is consistent with the literature, so we can really unambiguously assign that this is the sodium nitrate we're looking at. And again, the approach is in terms of quantifying the amount of either, in this case, nitrogen or oxygen atoms present, is to focus on the differences post-H minus the pre-H. So that gives you the quantitative measure of the number of atoms present. Here, and this is something that you will see all the time, and I would like to point it out. If you study using the AFAM, it gives you a very nice and direct way how you can get the three-dimensional morphology. So it gives you the way how you can assess the differences between thickness and diameter of size. And this is what we see for most of the samples. So note that the slope is always less than 1. So you can think of particles when they're deposited on the substrates, as Vicky likes to point out, like a pancakes. For us, however, this was very important because, again, the X-ray microscopy doesn't allow you a direct way how you can measure the height. But if you combine two methods together, that allows you to decouple the height. And so looking at the Beer's law, optical density is something that we measure. Height is something that we measure with the AFM. Mass absorption coefficient is tabulated values that we can put that are specific for different absorption ages for different types of atoms. And so that gives you now the fit, atomic absorbance as a function of thickness, in this case for oxygen, and nitrogen. And from the fit, we can determine the corresponding atomic densities, and we can get the atomic compositions. So in principle, also how it's changing is a function of size. But as you can see here, we have a very nice linear dependence, so size doesn't really matter. So in this particular cases, we have exactly the same response within this size range. So we can get the ratio, and we can also get the overall density. So I would like here one more time to point out that the density that we are measuring corresponds to the overall density that is equivalent to the volume occupied by the particles. So if you have any pores that are present within the particles, that would lead to the lowering of the density that we measure. Okay? So it gives you what I consider is a very nice way how we can quantify the extent of how porous the materials are. Limited, however, to carbon, oxygen, and nitrogen, at least so far. So we looked at more complex systems. So we looked at the mixtures. This is an example of looking at the malonic acid and sodium nitrate. So we see very nice homogeneous mixture in the AFM at carbon, nitrogen, oxygen maps. So we did exactly the same approach. So we collected the spectrum. We also determined what is the slope for the AFM measurements. And then we identified what is the atomic densities, carbon, nitrogen, and oxygen. So we can determine what is the atomic ratios in a quantitative manner for carbon, nitrogen, and oxygen. We can also compare it with the expected values. And as you can see, we have a very nice overlap between what we expect and what is present. And we can also quantify the total density, which would be the following number. So it also, as a side note, gives you the ability to predict what would be the ratio of components on the actual particle level relative to what it is in the solution. Because in some cases, and we see it in some systems, that ratio is different between the solution phase and what we have on the substrates. So the ability of looking at these ratios allows you to actually, in an independent way, how we can determine what is the ratio of components. And finally, this is a nice correlation with some of Alan's work. So as you can see, we have an example when we have a mixture, one-to-one mixture of butyric acid and sodium nitrate. And what we see, if you look just from the AFM perspective, so you see that they're nice and rounded, so it's very similar to before. But if you look at the phase, we see very clear differences. Now, phase in atomic force microscopy gives you some kind of chemical equivalent types of information. And so it tells us that there is some clear differences between how the material inside the core and outside interacts with our atomic force microscopy tube. Now, AFM does not really tell you what exactly we have. So we can suspect that one probably corresponds to inorganic component and another one to inorganic. And so this is where we turn to X-ray microscopy, which allows us to differentiate these two. And here is the examples of the spectrum and the corresponding maps at the carbon, nitrogen, which kind of got lost, I guess, and oxygen, where you can very clearly see, if you look at the carbon, that the shell, as we call it, consists primarily of acid and some inorganic component. The core consists primarily of inorganic components, sodium nitrate. What we also can do, we can go beyond that. So since we have the ability of combining AFM and the X-ray measurements, we can actually quantify. We can quantify what is the ratio of two components and how it changes as a function of both size of a particle and also how it changes depending if you're inside or outside of the particle. Sorry, if you're inside or outside of the core of the particle. And so if you look at the overall values, what you see here is the values for atomic carbon, nitrogen, and oxygen, and how it changes depending if you're in the core region, as we define it, or is in the shell region. And we have a very special analytical way how we can really differentiate and define what we call a core versus a shell. But it gives us the way to assess the ratios. So atomic ratios are given here from which we can also reconstruct the ratio of components. And as you can see, it gives us both means how we can quantify total density, and also gives us a way how we can quantify the ratio of the components. So in the core, we have the ratio of 1 to 5 between organic to inorganic, and in the shell, we have 1 to 0.65, approximately the ratio. And this is how it varies as a function of size. So what I illustrated, and here is an example that allows you, without a prior knowledge of what kind of particles you have, to determine the density of individual particles and determine atomic densities. And as I mentioned, assess the porosity of the materials. Okay, I think I'm going slower than I expected. So very quickly, so this is a very recent work, and I just would like to give you some very quick information about it. So this is the work that was done in collaboration with Laskin from Penn, and it's looking at the mixing states and chloride depletion when we have mixtures of sodium chloride with two organic acids, malonic acid and glutaric acid. And basically what we observe, and this is something that was observed previously, but here we are focusing on submicrometer particles, in that if you have aqueous solutions of a mixture of inorganic sodium chloride with either malonic or glutaric acid, so what we can observe is the following reaction and the formation of sodium malonate or sodium glutarate salts, and what is also important, the formation of HCl in gas phase that can be released, leading to the depletion of chloride within the particles. And so what we did, we utilized SEM EDX to probe such reactions on individual particle basis. And very briefly, here is the results. So this is sodium chloride, this is glutaric acid and malonic acid. So we see nice cubic structures for chloride and rounded for both acids. So when we start to mix them up, you see very interesting changes in terms of the mixing ratios. So as you increase the fractions, as you can see, we start to go from more cubical to more rounded structures. I would like to point out that 3 to 1 is a really nice homogeneous mixture. If you look at the glutaric and sodium chloride, somewhat similar effects are observed. And as you increase the concentration of organic, so you start to see very nice inclusions. And so for these samples, what we also did, we determined what is the mean elemental Cl to sodium ratio. And what is shown here is a function of particle size, the dependence just for pure sodium chloride. So 1 is the expected values. So you observe some decrease as you decrease the size, which is attributed to the beam damage. And now you compare it for the mixtures of glutaric acid with sodium chloride at 3 different fractions, 1 to 3, 1 to 1, and 3 to 1, and also malonic and sodium chloride. So basically, as you can see, you observe a very clear depletion of the ratio relative to what is expected of 1 for sodium chloride. So indeed, that reaction does occur, at least for these two low molecular weight decarboxylic acids. And we also observe that the extent of depletion clearly depends on the type of the acid. So observe significantly more depletion of chloride for malonic acid relative to glutaric acid. And the one that I pointed out, the 3 to 1 ratio, as you can see, leads to pretty much complete depletion. And so the particle therefore becomes really a salt now of organic acid. So this is, again, the work that was very recently published. And we also did the hygroscopic properties. I just don't have time to talk about that as well. But basically how we can explain the changes in hygroscopic properties with the changes that correspond to this reaction. So very quickly, finally, so this is ongoing work. I just literally have one slide as kind of the idea. So this is the work that we are doing. And we recently joined the case. And so some of the methods that we developed, we are going to apply to a variety of different problems that both Kim and Vicky very nicely explained. And so this is one of the experimental ideas that I just would like to point out that we have it and it appears to be working, giving us a very nice results. And the idea is to use atomic force microscopy as means to determine surface tension of individual droplets. So this surface tension is important, for example, in terms of determining the viscosity of different systems or how liquid-like the materials are. And we can do these measurements under different relative humidities. And so here's an example of if you raise the relative humidity, again, we can also look at the changes in the phase transitions and study the hygroscopic properties and so on. So the idea is very simple. The only thing is that you need to have a very specific what we call nanoneedles that have a cylindrical shape. They have 50 nanometers in diameter. And the idea is that you come then in the contact with the particles. So as you can imagine, you need to have a corresponding ratio between the size of a tip relative to size of a particle. So you're talking probably half a micron, maybe several hundred nanometers. So this is our limitation at the moment. And so what is shown here is a typical force plot. So looking at the force as a function of Z sensors, which basically brings the tip in and out of the contact. And so as you go in, at some point you touch the droplet, it drops in, you have a very nice constant force, and then you start to touch the substrate, so the force increases very dramatically. Then you reverse the direction, and you observe the retract from the surface. And basically, what you can measure, and as you can see, you can measure in a very clear way what we call the retention force or adhesion force. And for well-defined geometry, such as this cylindrical tip, we can relate the measured retention force to the surface tension, knowing the size of the needles. And so by doing that, here is an example of looking at the glutaric acid particles as a function of relative humidity, where we observe the dependence, which is consistent with the literature values. And so it appears to be working, and it looks like it's a really nice technique, which we hope to push on and study a variety of problems. I'm out of time. So just very quickly, in summary, I just give you a few examples where we developed a variety of analytical tools, primarily utilizing X-ray microscopy and atomic force microscopy to study the variety of these problems. And I will finish with thank you for coming, and I would like to acknowledge all the people who really helped, and in particular Vicky Gracia and Betsy Stone. So we are the last couple of slides were really related to the work that we are doing at the moment. Thank you very much.
Questions? Alan?
The last topic, you talked about surface tension.
Yeah.
When you pull the needle through the particle, can you also get the viscosity out of that curve as well?
Potentially, yes. 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 For that, I would argue it probably will be better to use different types of tips, and we use that. But for pulling away, these types of tips are much better. But for going in, so we also have a conical tip type of shapes. And so this is something that we do, yes. And in fact, a lot of research that we do in my group is actually related to measuring how hard or soft materials are. So we have a really good handle how we can quantify the mechanical properties when we actually push in. Yeah.
This is really nice. It's the relation between the height and the diameter, that 0.29 or whatever that is. Do you get other information from that, or how much does that vary? I was actually surprised it was so linear across the entire size regime.
It is. Well,
Do you see variations in that when you change the relative humidity space or other types of what? That seems like the slope of that function tells.
that slope tells us, I think, it relates to the viscosity or how glassy the materials are. So this is one way how we're kind of thinking of using this information as a quantitative or semi-quantitative way how we can determine that. Now, in terms of as a function of relative humidity, so all these measurements, so when we were showing those plots, they were under dry conditions. So I guess I do not know how it changes as you raise the relative humidity. That's something that we can definitely take a look at. Yeah.
Any other questions? Okay, I think we'll move on. Thank you very
Thank
much.
you.